All right. This is a really exciting seminar for me personally because Dr. Glenn Gaudette is a great friend of GFI's. He's a remarkable scientist and ecosystem builder, and has really done a lot to advance our field in the relatively short amount of time that it's been in existence. So we're so thrilled to have Glenn with us here today and so excited to learn from him. Um, Dr. Glenn Gaudette is the inaugural chair and professor of engineering at Boston College. He and his collaborators at Boston College and back at Worcester Polytechnic Institute have pioneered the use of plants as scaffolds for heart regeneration. And this work has led to really significant recognition, including his work and his colleagues and his lab being featured in media outlets around the world. And now this really impressive work is being used to power innovation in the cultivated meat field. So this is a really great example of the kind of interdisciplinary nature of our field. Glenn today will be diving into developing edible scaffolds for cultivated meat production, utilizing vegetables such as spinach and broccoli florets, so that we can tap into the really powerful vasculature that's used to transport fluid throughout tissue. So without further ado, Glenn, I will pass it off to you to share your screen and get us started with the seminar today. Thank you again for joining. And I can't thank you enough for the successes and milestones you've helped our field hit in recent years.
Thanks, Amy. And yeah, thank you for that kind introduction. And thank you for everybody attending today. And I owe a lot of thanks to Amy and GFI also, and I'll explain a little bit of that in just a minute. So today I want to talk a little bit about some of our work on decellularized spinach and some other plants that we've used, and tell you a little bit about how I got to this field and where we are and where we hope to go from here. So to start off with, tell you a little bit about my journey in cellular agriculture. It really all started by taking the train. And so when I was invited to give a lecture at Cardiff University back in December of 2017, and my good friend and colleague Marianne Ellis is at the University of Bath and was kind of on the way. And so I took the train from London out there to Cardiff and stopped off and had lunch with Marianne, and she first introduced me to her work that she was doing on lab-grown meat, cultured meat. You know, we didn't even know what to call it. You know, then it was still, the field was still starting out, and it really got me excited about it. And when I got back to Worcester, I was at Worcester Polytechnic at the time, and Amy, great job pronouncing Worcester. Some people have a hard time pronouncing Worcester, but as I always say, you just say it just like it's spelled, Worcester. So Worcester Polytechnic Institute. I got back there and I started digging into literature and found this great article by Liz Speck there at GFI on opportunities really in the field, in the clean meat industry. And then shortly after that, I actually got invited by Liz to give a talk at their annual meeting, the GFI meeting. And that really got me thinking more about this field. And I don't know, a couple months later, Paul Shapiro's book on clean meat came out, and he sent me a copy with a nice little note on the inside of it, which was reinvigorating to me. And I knew, this is something that I really, wanted to get into. And so that's really how my interest in the field, got started. And so why was I traveling to Cardiff? I was going there to talk a little bit about our work in tissue engineering. I spent a couple of slides just to start off with, give you some background on what we were doing at the time. We're still doing, but a large percentage of our lab really is focused more onto cellular agriculture. I'm not going to bore you with all the details of the slide, but really what I want you to focus on is this pink area on the right, because that pink area represents the unmet need, unfortunately, of patients that need an organ transplant. And so, and as you can see, this is a few years old, but this pink area here is the difference between the number of organs that are needed, over 122,000, and the number of organs that are available for transplantation, a little less than 31,000. And so we were looking to try to address this problem, and we were more focused on cardiac tissue engineering and cardiac regeneration. So we were growing human heart muscle cells on fibrin sutures. And so what you see here is a fibrin suture contracting. You see a microscopic view of that here. Down below, this is represents the electrical activity in the cells. And so we were able to essentially grow human heart cells, tissue perhaps, on these fibrin sutures. Each one of these sutures is approximately 100 microns in diameter. And so if you think about what we really need and what the human need is, it's a thick muscle, kind of like when we think about cellular agriculture. What part of what we need are thick cuts, steaks, right, to replace what's currently being eaten out there and consumed. And so if we stack a bunch of these sutures on top of each other, we might be able to get to about 400 microns in thickness. But then the problem becomes an issue of oxygen diffusion into the cells. And so there's something that's known as a diffusion limit. And after about 100 to 200 microns, oxygen can just not diffuse into most tissue. And so that means that the cells that are inside there are beyond 100 to 200 microns of a blood source are going to die. And so this is something we needed to overcome. And one of the things we were looking at the time was decellularization. Decellularization is essentially a process where we wash out the cells, but we leave the structure of the organ or the tissue behind. And in this case here, we're looking at heart tissue. And so this image is an image from a paper that Harold Ott did a few years ago, where he decellularized the rat heart and left the structure behind. And the thought here is we could then repopulate that tissue with human heart muscle cells that could then contract. And I was fortunate enough to spend a sabbatical with Harold in his lab, which is now down the road here at Mass General Hospital, where we actually did something similar with human hearts. And we decellularized human hearts that were rejected for transplant. And again, our hope was to repopulate that scaffold with human heart muscle cells that contracted. It takes about 5 billion cells to do that. We were able to demonstrate that we could actually repopulate a small portion of that decellularized human heart, and that it would start contracting again. But these hearts are in short supply, they're very expensive, and they're very variable from person to person. So we started thinking, can we find a more sustainable and controllable supply? And where to look? Well, at your salad plate, right? So we looked at spinach. And when you look closely at spinach, perhaps you'll see the veins in that leaf, and you'll see the branching structure. And so that's what we looked at. We took a look at the spinach leaf, and we compared it to, in this case here, the decellularized rat heart. And what hopefully you can see is this branching pattern that we have in our bodies, in our blood vessels. And it's very similar to the branching pattern that we see in plants. So in green on the right is put the laser pointer on. Here in green are the veins of in this case here, the leaf that's been decellularized. And you can see hopefully the branching structure on the leaf there, as you can see in the rat heart here in the arteries in the rat. And so it turns out that plants need to transport fluid, just like our body does. And those plant cells also need to stay alive. And so we then looked to decellularize the spinach leaf. And we based this off the way we were decellularizing rat and human hearts at the time, which is put a tube or a catheter essentially inside the aorta of the rat heart, and then we perfuse it, and so the detergent goes throughout the entire heart and removes the heart cells. My graduate student, Josh Gerschlach, when he saw this spinach leaf, he thought this end of the spinach leaf here, the pedicle, reminded him of an aorta. And so when you're a cardiac surgeon, or a cardiac researcher, everything looks like a heart to you. And so he thought the spinach leaf resembled the heart and stuck a tube and a needle inside the end of the leaf. And that was the way we originally started to decellularize spinach leaves. And so in order to do this, we put a solution up high in the air. And so we use a process called gravity drain. So for the engineers out there, you're probably familiar with hydrostatic pressure and things like that. And so it's a very cheap and easy way to perfuse, in this case here, a leaf. And so we would cannulate the leaf, which means put a tube in the end of the leaf. We'd wash it with hexanes to get rid of the glossy, the cuticle layer, the waxy layer on the outside of the leaf. And then we would perfuse the leaf with SDS, which is essentially a soap, a detergent, Triton X, which is another soap or detergent, and 10% bleach. And then we would wash it for about a day with deionized water, distilled deionized water. And then we could either store it lyophilized, and so we could essentially freeze dry it, take all the water out of it so it's nice and light, and store it that way, or we could leave it in water until we were ready to use it. And on the right of this image of this slide, you see the setup. It's kind of cut in the middle here, but the tube is up in the ear. It comes down and attaches to the leaf, which is down here, and it's blown up on the right. And what does this look like? Well, if we look at it over in a time-lapse photography, and that's what this here is time-lapse over about 5 days, you can see hopefully the spinach leaf losing that deep green color. And now you can see the cells actually getting all washed away, and it's transparent, and you can actually see right through the leaf. But you can still see those vessels inside that leaf. And so when we look at it over time, this is essentially how the leaf changes from day 0 to day 1, day 5, and day 7, where now you can see right through that leaf at day 7. In working together with Bill Murphy in his lab at the University of Wisconsin, we did some histology and some scanning electron micrographs. On the left is some histology. And so if we look at the native spinach leaf before it's been decellularized, we see a bunch of cells, and these blue dots kind of represent those cells. And then once we take them out, we decellularize it, we can see that there are no more blue dots, but we still see the veins that exist. And so these kind of dark red patterns here and kind of blown up here, those are the spinach leaf veins. And we showed that we're able to remove the DNA from the leaf and remove a lot of the protein, but some of the protein stays behind. This could be a plus or a minus, but we look at that as an opportunity. And up at the top right, you see the non-decellularized leaf, an electron micrograph of it, and then down below after it's been decellularized. And of course, the big question for us was, is this still, are these veins still open? Can we still perfuse? And in this case here, we did red dye, but we've also done blood, and it still perfuses through the leaf. And so those veins are still open and allow flow to go through there. In this case here, again, red dye. In addition, we seeded human heart muscle cells onto the leaf. And so hopefully you can see in this region here, the cells that are actually moving. You can see them contracting. In the background here, I'm putting the cursor over it, the pointer, is the actual leaf vein. And you can see that there's a close interaction between these cells and the leaf vein, and more over here. And so we're actually able to put these human heart muscle cells on the leaf and actually get the cells to contract. On the right-hand side is just a chemistry image showing some of the structure of those cells and the striation that we like to see in muscle cells that we could see here. So that was pretty exciting to us. But then I took this trip and got this invitation from GFI and started learning about actually bigger problems. And I'm a mechanical engineer, is my background, biomedical engineering, and I think I can make I hope to make a difference to the world. But once I found out about animal agriculture and the opportunities for growing meat and not needing to sacrifice the animals, not needing to use all the land that the animals require, not needing to use all the water that the animals require. I thought, could I really make a difference in our world and actually help our environment and help people? And that's when I started getting more involved into cellular agriculture and cultured meat. And so I'm not going to go over all the numbers. I'm sure you folks know these numbers just as well as I do. The bottom line is we can and we must do better. And one of the things to me that has really been amazing about this field is the fact that people are working together collaboratively. And it's really great to be part of this. It doesn't matter what your background is. Everybody can contribute to this field. And that's truly the way I feel. And I've been very fortunate to be part of this. And being part of the field, I've been able to meet folks from so many different backgrounds. It's really been fantastic for me. So when we think about, one part of this solution, it's really making the meat. How do we make cultivated meat? And what does it take to make cultivated meat? Well, simply it takes cells, takes solution or media to grow them in, and generally it takes a scaffold to grow them on. We put them all together in a bioreactor, and out the other side we get edible meat. Sounds pretty simple and straightforward. And of course, nothing in life is ever that easy, right? So it's much more complicated, and there's a lot of nuances to all these different pieces. And one of the other really, really important pieces that we need to consider is regulatory. So when we're thinking about bioreactors, for example, there are many, many different questions we can ask. Is it a perfusion bioreactor, hollow tube bioreactor? A lot of different types of bioreactors we can consider. We've got to think about the filtration. How are we going to change the media? How are we going to filter it? How are we going to control the growth rate? And what does that mean for our bioreactor? In regards to the cells, what source are we going to use? We can think about using many different types of cells from many different animals. We can think about using induced pluripotent stem cells versus primary satellite cells. What about fat cells versus precursor cells for skeletal myocytes? We've got to think about how we're going to grow them, how we're going to proliferate them. And then we've got to think about how we're going to differentiate them, because generally cells are either going to proliferate or they're going to differentiate, but it's one or the other generally. And so once a cell starts to differentiate, it's hard to get it to proliferate again. So generally we want to proliferate them, start off with a few cells, 100,000 cells, 1,000,000 cells, and continue to double them, and then have some of them differentiate and turn into the muscle cells. Generally that means different states in those cells. And then how are we going to combine different types of cells? What if we want fat cells in there? We want muscle cells in there. We've got to think about how we're going to grow these together. Regulatory is an evolving process. We know that the FDA and USDA is going to regulate lab-grown meat, cultured meat in the United States. What does that mean? How are they going to regulate it? Those are questions that we're still seeking clarification on and some clarity. But we can work with our regulatory agencies. And I know GFI has done that and continues to try to do that. I think that's really, really important. We can't forget the importance of the regulatory boards here, in the U.S., whether it's the U.S. Congress or even the state legislature, because they can actually pass laws that will make cultured meat. It'll increase the hurdles for cultured meat to actually get to the market. And so we want to be careful. We want to work with them, and we want to embrace them really, and help them see the benefits of what we're trying to do. In regards to the media, a lot of different things to consider. Serum, how much serum, where we're going to get the serum from. Right now, we generally get it from animals. We don't want to get it from animals in the future. That generally, and there is some serum-free solutions out there, but that generally increases the cost. Growth factors, where are we going to get our growth factors from? Again, they're very expensive. Scaffolds. Do we want to use a vascularized scaffold? Is it a perfusible scaffold? What's the cost associated with that? Do we want to look at edible scaffolds? Do we want to look at degradable scaffolds? There are a lot of different factors to think about when we're looking at this entire process. In regards to our scaffold that I told you about before, we were using decellularized spinach. And this is the process that I showed you earlier. Well, if you think for a minute about how are we going to translate this into a commercial reality, right? And so as an engineer, quite frequently, we think about these, great ideas, but then we think, how are we going to scale that and meet the big need that's out there? And when we start thinking about this process, one of the first things I think about is the cannulation of the spinach leaf. It's a time-intensive process that's very delicate. We've got to do away with this. This is going to drive up cost. Right? And so we want to keep everything as cheap as possible. And so we've moved away from that, and I'll show you in a minute what we're doing with that there. We used hexanes to wash off the glossy, the cuticle, the waxy layer on the leaf. Hexanes are essentially a gasoline. It's essentially like gasoline. We don't want to douse our food with gasoline before we eat it. So we've got to get rid of the hexanes. And it turns out we can just extend the time that we decellularize the material for, and we can get rid of the hexane. And also, Triton X is not something that people in the industry are familiar with. So we've moved on from this and looked to streamline the process essentially. Instead of cannulating every single leaf now, we actually put them essentially in a beaker, a 1 liter, 2 liter beaker. We use SDS for 2 days. And then instead of using Triton X 100, we use Tween 20 now. Tween 20 is a detergent or ingredient that's found in cough drops in other food already. So it's approved to be, as an edible ingredient. And we also use, we had bleach. We use the bleach tablets that are used if you ever go hiking and you want to make sure the water that you take from the stream is safe. They make tablets that you can just drop in there. And so we use a similar tablet in regards to our process. And then we wash the leaves in distilled deionized water afterward. And so down below here, you can see, and if you look closely, hopefully you can see the green leaves right here. After a couple of days, they start sinking a little bit more. And I guess I should point out on the bottom here, we put a metal grate. You can see it more on the right with a stir bar underneath that. And over time, and you can see in this third image here in the beaker, those leaves become decellularized. And so again, the important thing about this here is we're trying to make the process more streamlined and scale it up. And so this is a 1 liter beaker. Can we do this with a 55-gallon drum? Can we do this with something even larger? I hope so, but we haven't done it yet. So now that we've essentially decellularized the spinach leaf, we wanted to see if we could actually grow some cells on these leaves. And our process for this is we start off by taking a small biopsy. And so one of the things we've done working with our colleagues at Tufts and David Kaplan, they go out to the Tufts vet school and we'll take a small biopsy of a cow, small muscle biopsy, and then sew the cow up and send the cow back in the field. And so we could take those cells, those are primary cells. We could isolate the satellite cells, which are the myocyte precursor cells, so the muscle precursor cells, and then expand them or allow them to proliferate. And then once they've proliferated, we can then add them to the decellularized spinach scaffold and see if they'll actually adhere to the scaffold. And hopefully they'll initially proliferate and then differentiate and fuse and form a muscle fiber. So the first thing we sought to do was see if we could actually isolate these cells, grow them up in a dish, and get them to actually contract. And so if you look closely here, and you see it probably best at the very tip here, you can see that cell is actually contracting. So these are the cells that we isolated the way with the process I just showed you, grew them up in culture, and can actually get them to contract. So how often do we want to contract these cells? We don't know. What frequency? Do we want to contract them 1 hour a day? Do we want to contract them every second? All this stuff, we don't know yet. A lot of work left to be done in the field. What makes for the best tasting meat once we've grown it? We've got a lot of work left to do. We put these isolated satellite cells on spinach, and on the left image here, we stain these here for F-actin with fluoridin, and so that's a green stain, and the blue stain is the nuclei. And what you can see are a whole bunch of cells here on a spinach leaf. Again, the blue dots represent the nuclei, and so in some cases here they're aligning, and so they're probably fusing together here. In order to determine if they're actually turning into muscle cells, we stain for myotubes, and that's shown in green. And so here you can see this long, thin cell with the blue nuclei inside of it. And so that there's a nice long muscle fiber that's forming on top of the spinach leaf. And again, just some more examples on the right-hand side. One of the things when you think about when you think about meat, you think about the structure and the organization of the fibers. And so one of the things we wanted to test is, are these fibers actually aligning? And so we started off looking at growing these cells just on a normal tissue culture plate in culture. And what my student Jordan Jones did was he looked at the frequency of distribution. So what's the primary angle that these cells are at? And in this case here in the control, they're pretty much all over the place. And yet what you see in cow number one, and so these are 3 different cows that Jordan took samples from, a pretty nice alignment. And so you can see the green is all pointing in the same direction. And so when Jordan analyzes this, what you see is a nice sharp peak in the distribution right here. And so most of these cells are aligned, in this case here, around 45 degrees, a little bit more than that. The key thing is not the angle, but the key thing is the distribution and how tight that is. And so a lot of these cells are all oriented in the same direction, which again is going to be important if you want to have a fiber direction. A different cow had a similar distribution, yet another cow, it's all over the place. And so we don't know, are there specific samples that align better than others? Did we do something differently? Was it the spinach leaf? We don't know yet. This is, area for further investigation. But the good news is it kind of shows that it might be possible to actually align these cells on their own on the leaf. Of course, why stop at spinach? There's so many other wonderful plants that we could think about. We looked at lettuce, and so here are some cells growing on lettuce. Again, the green represents the F-actin, the fluoridin staining. The blue are the nuclei. We've also grown satellite cells on jackfruit, on decellularized jackfruit. And so on the left, again, the blue represents the nuclei. The green is the fluoridin staining, the F-actin structure. A higher magnification, you can see the kind of fiber direction, and also more images here. And so jackfruit provides a nice fiber orientation that's inherent to it, it's a part of the fruit. And so we're hoping to use that fiber that's present already to help provide texture to the meat when it's grown. And one of more recent things we've tried is broccoli florets. And so this is the very tip of the broccoli, which we can, easily cut off and then decellularize and then actually grow these satellite cells. So again, you see the satellite cells here. In the background, you can see some of the veins also in the broccoli. So, if we really want to scale this up, though, we need to start thinking again about how can we move this to a more user-friendly, more simple process. And so what a group of students in my lab did about a year ago, just before COVID hit, they took isolated cells from the cow and they placed them in a 15 ml conical tube, and they put 6 milliliters of media in there, and they put 100,000 bovine cells, the satellite cells, and a few pieces of decellularized spinach. And what they did was they just put this together in the beaker, in the cylindrical tube, in the conical tube. And so normally what we do is we take a spinach leaf and we pipette the cells on top of them. But again, thinking about getting more user-friendly, a little easier, what we did was we just added them all together in a conical tube and said, if we just put them in together, will they find each other? And we then stained these, and here's some images from 2 days later. And so again, somehow the cells, the cells are floating together in the space and slowly agitate them. And so they come in contact with the leaves, they anchor on the leaves, and then they start proliferating on top of the leaf. And so after 2 days, we were able to get a near confluent layer on those decellularized spinach leaves. And so you can see here side A, but also the cells adhere to the other side, to the back side. And so we were able to get cells on both sides. And again, that's pretty exciting for us because if we want to think about making it easier, we don't need to pipette anymore. We still need to figure out how many cells are there, so we still need to pipette. But we just need to put them into a conical tube with media, with decellularized scaffolds, and they're going to grow on those scaffolds. The next question we asked, though, is what if we added more blank scaffolds? Would they actually go from one scaffold to the next? And what my students did here was they said, on day 0, so when we first throw things together, what if we put circular pieces of the decellularized spinach leaf in? We let them sit for 2 days, and then after 2 days, we take off the media and we replace it with fresh media. And when we take off the media, of course, any cells that are not adhered are also going to come out with that. And then we throw in some blank scaffolds, some decellularized scaffolds, but this time they cut them in the shape of a triangle. And they put them in there. And then 2 days later, they changed the media again, and they put in square scaffolds into the conical tube. And then we waited a couple more days, and then we analyzed those. And we said, okay, well, what's happening? Are the cells actually jumping from one scaffold to the next? And what you can see is on the circular scaffolds here, again, the blue dots representing the nuclei, the green representing the actin structure, and so the presence of cells here, the presence of cells on the triangular scaffolds, the presence of cells on the square scaffolds, more cells on the scaffolds we threw in at day 0 compared to day 4, but clearly the cells were present on all 3 scaffolds, all 3 different shapes of scaffold, kind of demonstrating the transfer from one scaffold to the next. And so how are we going to take advantage of this? Well, we're currently working on a bioreactor system where we have a proliferation reservoir, which is shown here on the left schematically, where now we can put in the primer cells in the plant-based scaffolds at T = 0, just like we did with the 15 ml conical tube, except we can scale it up a little bit, hopefully. And we can allow the cells to proliferate on the scaffold and add more blank scaffold over time. And then we can cycle them through a side port where we can then analyze them as they're coming through and use sensors to determine if they're confluent or not. Do we have enough cells on the scaffold? And if the answer is no, they'll just get recirculated back and put back into the diffusion reactor. If the answer is yes, then they get passed on to the differentiation reactor. And so now they can be exposed to different chemicals. We can add growth factors in there, or take away serum. And we can have another side port here, and this side port here, in addition to looking at the cells, we can also electrically stimulate them as they're passing through. And we can actually see, are they contracting? And if they are they contracting enough? If they're not, we can again pass them back through and put them back into the differentiation reactor. And if they are, we can pass them on, and now we can send them on to packaging. So this is our long-term goal. We're not there yet, but we're working on that. And so why do we think this might work? Well, going back to some of the data I just showed you, we want to start off with taking the primary cells from an animal source, putting them together. We've shown you that, shown you recently, in the previous slides, that grow on spinach, broccoli florets. We kind of like broccoli florets because they remind us a little bit of the macrocarriers that are current, or microcarriers that are currently used with these bioreactors. And so we've shown that they adhere to them. We've shown that they'll actually grow and proliferate on those scaffolds. We know that we could just add additional blank scaffolds, and they'll proliferate on those scaffolds, or they'll leave one scaffold and attach to the new blank scaffold. And this is some recent work my student did, where we kind of tried to make this side loop. Let me just play this movie for you on the bottom right. If you look closely there, you can see that clear tube, and flowing through there are these decellularized broccoli florets. And so I got it kind of blown up. I'll play it again. That part of that blown up on the bottom left, but those are actually small pieces of broccoli that have been decellularized. We haven't loaded cells on them yet, but they're actually passing through the side port, demonstrating that we can actually use this side port system. And our plan is to use an optical sensor or camera system where we can electrically stimulate the cells as they pass through here and actually image them and see how they actually contract. So, we still have many, problems that limit the field. We're trying to overcome some, and there's still a lot more, but, the technical acuity required to do this, currently, a lot of the companies are looking for students, they're looking for graduates with technical degrees that know how to do a lot of, high-level biology type work. And so that requires a high level of, training. Can we get away from that a little bit? So we hope that this type of a process will help get us away from that at least a little bit. It's very expensive in terms of the media, the cells, the scaffolds, the labor. Making things more user-friendly, reducing the user input can hopefully reduce some of those costs. Consumer acceptance, of course, is still, we're not sure if the consumer will accept it. And some of the studies, I've looked at, and my students did a small study, suggests that the younger generation is much more interested in cultured meat. My generation, probably about 50-50. But I think one of the key things here is we've really got to think about how do we ensure that the meat eaters are willing to eat this meat, right? And so a lot of times people ask me, is this something vegans might be able to, might be interested in eating? That's totally up to them. Some of the folks I've talked to said yes, some have said no. But if we really, really want to make a difference in our world, for our environment, we've got to make sure that we take the meat eaters, right, and make sure that they are very interested in taking this and eating this product, because they're the ones who, are currently, and I'm a meat eater myself, and so, how do we get people to say, I want to try this? And I enjoy the Impossible Burger. I enjoy the, all these plant alternatives. They're getting much, much better than they were 20 years ago. So we've got to continue that. And I think, that's part of what we need to focus on with the consumer acceptance. We've got to think about the regulatory approval process too, and that's the FDA, USDA, and so having a system like a bioreactor system like this, we're hoping that that'll provide us the opportunity for sensors. So what's next from a technical point of view? There's a lot left to be done here, right? So there's work for everybody, right? There's the cells, the donor, what type of cell, what is a doubling time? Can we increase it? What type of cell? Differentiation capabilities. There's the scaffold. I talked about one scaffold. There's a lot of other promising scaffolds out there. I think, and I hope eventually there's going to be several different alternatives, because just like today, there's so many different meat products out there. Can we have different products, different cell types, different scaffold types, and maybe even grown in different media? There's a lot of opportunities out there. So there's a lot of work left to be done in this area here. But one of the other areas that I'm really excited about is the collaborations. And recently, a team that I was part of submitted a IUCRC, an Industry University Cooperative Research Center proposal. We're very hopeful that the NSF funds this. The PI of that project is Tanya Domingo, my colleague and friend at Worcester Polytechnic Institute. Bill Murphy and Greg Ziegler are both site PIs on that from the University of Wisconsin-Madison and State University. And that really looks to bring together folks from industry, different companies, in academia, and try to do pre-competitive research to really help the field move forward. I'm really, really excited about that. I really hope it gets funded, because I think that this can really help push the field forward. And the other thing I'm excited about is the collaborations. It really goes beyond engineering. If you had asked me 10 years ago, 15 years ago, what collaboration looks like, I might have said, well, as a mechanical engineer, I could work with an electrical engineer. Those are two totally different fields. And, that's collaborations for me, in terms of multidisciplinary collaborations. We need to go much, much further than that. Social scientists, food scientists, environmentalists, political scientists, philosophers, theologists, economists, anybody who wants to make the world a better place, we need to embrace them and tell them there's work here to be done. There's so much in this area that can be done. And so what's next in regards for me, and if some of you may be familiar, I may know some of you and know me from WPI, Worcester Polytechnic Institute. I spent 15 years there. Really, really enjoyed my time there. But I'm really excited to be at Boston College, where we're building an engineering program. Boston College never had any engineering before. So we're really excited about that. We're enrolling our first students in the fall. And we're really doing more than just that technical knowledge. What I'm excited about in engineering education is providing students with the technical knowledge that they need to be engineers, but with a mindset focused on helping others. And how do they get that mindset? They get that mindset by working with people from different fields and really being multidisciplinary, convergence research. It's so important. It's so important for our society, just in everyday life. And it's so important for science and engineering to have a multidisciplinary background, right? In multi-ethnical background, multi-socioeconomical background, multidisciplinary background. Diversity is really, really important in our field in many, many different ways. And I'm excited about that opportunity at Boston College. Of course, none of this work was done on my own. As I like to tell my students, they do all the hard work, and I go out and give all the nice presentations and get all the credit for it. But really, it's their work. And in some of my faculty collaborators are listed here. I'm not going to spend the time to go over all of them. I've been able to mention a few of them during the talk. And the students are really the important ones. They do so much work here. They're the reason that I'm able to present any of this work. And to be honest with you, they're the ones who come up with all the great ideas. We work together, we treat each other as peers, and I think that's really, really important. I really, I try to encourage an atmosphere where everybody feels welcomed, everybody feels that their input is valued. That's the kind of environment I think that's needed in science going forward. So I try to do that, and I hope I'm successful there. And happy to take any questions you might have. A little cartoon that my students found or made or something that I like to end my talks with. Thank you.
That's an awesome cartoon, Glenn. Thank you so much for this phenomenal seminar. I feel so re-energized about the opportunities in this field. And just thank you so much for taking the time to share all of your exciting work with us. Awesome. Well, now we can move on to the Q&A portion of our seminar today. Those of you who haven't already gotten the chance to do this, please do feel free to drop any questions you have for Glenn about his work or future directions of his work there in the Q&A section, and we'll work down the list. Awesome. So let me just quickly pull up our Q&A. All right, Dr. Glenn Gaudette, the first question for you is, what do you consider the relative advantages and disadvantages of this decellularized plant material approach compared to other scaffolds for cultivated meat, like nanofibers, mycelium, hydrogels, or other approaches?
I think each approach has their own advantages and disadvantages. I think the approach that we use with the decellularized leaf, keeping that vascular network in place is going to be important. I focused on the bioreactor towards the end of our talk, but imagine if we took spinach leaves that were perfused and we actually stacked them on top of each other. We could think about creating a thick cut of meat that way. And having that perfusion and being able to basically perfuse it as we're stacking them on top of each other to keep those cells alive, I think is going to be an advantage. In addition, I think things like jackfruit have this structure already inherent to it that's really going to help the taste and the texture of it. So I think there are advantages. There are disadvantages too. It's an extra processing step. There's differences between a spinach leaf grown in California compared to Massachusetts, right? So there are a lot of variability also. In regards to the other techniques, I think that they all have their benefits. We've worked some with mycelium. Very high on mycelium. I think there's a lot of opportunities there. The hydrogels. Depending on if you're looking at the hydrogels for protecting the cells as they're exposed to a shear stress in a bioreactor, that's huge advantage. But there are also disadvantages, and so I think for the field, I hope we move forward with all of them because I think there's going to be opportunities for all of them too. And you think about all the different types of meat products that are out there: sausage, a chicken nugget, a fish cake. Maybe mycelium's best for one, and decellularized broccoli is better for another. Hydrogels are yet better for another one. And so I think there are opportunities for all of them out there.
Awesome. Thank you so much for your response. Yeah, I think you're probably right that one of the big benefits of using decellularized plant materials is maybe lower barriers of access and working through the regulatory process, because these are already kind of edible materials that have been used widely throughout the food industry. Yeah, very, very cool. The next question here is about whether the spinach leaves are coated with lamin or an equivalent before you start to seed the bovine satellite cells onto them.
So we have coated in laminin and collagen, but in this case here, the data I showed was not coated. They were not uncoated. And so in some cases, depending on the cell type, coating the leaf helps. In other cases, it really doesn't seem to make a difference. So in this case here, we did not coat them, but they could be. But we're also looking for a coating that is, edible, that the consumer is familiar with. And so I think there may be opportunities there for different coats, different coatings.
Awesome, thank you. A question here from Ivana from the University of Novi Sad or the Biosense Institute, praising you for your really great talk and then asking a series of questions. The first is whether you seed the cells only onto the surface of the leaf or also in the veins.
Only onto the surface. In our work in cardiac, we did seed endothelial cells inside the veins, but on our work here with cultured meat, we're just putting them on the outside.
Cool. The second question is what the myoblasts are actually aligning to. Is it the cellulose fibers of the leaf?
Yeah, we think it is. We haven't concluded that. We haven't done the studies to clearly show that, but we do believe it. And so Bill Murphy of Wisconsin, what he's done is he's done some really nice SEMs on the structure, and you can see that different leaves have different patterns on there, and the cells will align to that structure. We haven't done that study with these cells on this type of decelled leaf, but we think that that's what's happening.
Very cool. Yeah, she pairs this with a suggestion that asks whether you'd consider trying asparagus because it's so, fibrous or fibrillar.
No, because I like to eat asparagus too much, so I haven't tried it. No, we haven't tried asparagus yet.
Another question here is about what an actual scale-up pathway might look like. So how might you actually imagine making a three-dimensional whole cut-like construct from these really thin plant materials?
Yeah, I could envision stacking the leaves on top of each other and really, and then cutting it and on face at the end, because you can imagine stacking 4 leaves that are grown with muscle cells essentially on them, and then a couple with fat cells on them, and then more muscle. And so then when you look at it on face, on the end of it, you could see muscle fat, and you can see that kind of marbling structure. Perhaps that's a whole.
Yeah, super cool. Yeah, especially with spinach, it seems like you could make very precise marbling structures because of just how thin it is. That's a very cool idea. And the last question from Ivana is whether you see these kind of leafy carriers floating well in suspension. I know you were talking towards the end about, the entire bioprocess development that your team is now exploring. Do you see them behaving mostly as microcarriers?
Yeah, the broccoli florets especially seem to behave pretty well as microcarriers. The spinach leaves, we haven't actually used them in a kind of a spinner flask. We've only used them as a shaker in a shaker flask. And they move well in the shaker flask, but you'd expect that because of that setup.
Yeah. Very cool. This question here is about, maybe the end product formulation stage, or just helping us understand how well the scaffolds actually perform in a culinary environment. So have you tried to cook the recellularized spinach, broccoli, jackfruit, etc., after inducing this kind of differentiation?
No, not yet. Not yet. But my Ph.D. student, Jordan Jones, says he's not going to graduate until he can actually do that and serve that at his Ph.D. defense. And so,
That's awesome. What a commitment. Yeah, maybe we can invite you back for a live cooking demo.
Yeah.
See how they perform over heat. Awesome. This next question is kind of about the techno-economics of this approach to scaffolding. Do you have any general sense of the potential cost of what decellularized plant matter as a scaffold would cost per kilogram or per pound of meat over the long term?
So we haven't done that calculation yet, but all the the ingredients we use for the decellularization are all very low cost, and so the SDS is very cheap. The tween 20 is very cheap. Bleach in water, really, that's all it takes for the decellularization process, and then time. And so we haven't done the exact calculation though, but I'm a cheap person, and so all these things are really cheap, and you could see the setup is really, the original setup we use is really cheap, but even now it's so much easier to do it in just store-bought.
Yeah, I really appreciated those slides when you kind of X'd out the things that were obviously going to be massive hurdles to scale up, and tried to remove those from the process. Also, just generally appreciated this focus on the user experience. And ultimately, like, the things that gain traction are the ones that are kind of intuitive to use, or, relatively simple to adopt. So I think,
One of the things my colleague at Penn State, Rob Childs, is really suggested and stuck with me is we want to be able to bring along the farmers and the ranchers, the folks that are already in the animal agriculture industry. And so, can we make this user friendly so that they see this as something they can add, to their farm, essentially, in a new revenue stream for them, so that they can really decrease their herd and yet not decrease the amount of money that they're bringing in, right? And so, the economical model is really important too. We don't want to, we want to, see if we can bring everybody along, including our
yeah,
existing.
yeah, and it seems like there's this massive opportunity to just decentralize food production and bolster the kind of resilience of our food system by doing so. So, like, could the average citizen be involved in meat production in a way that they really just aren't in industrialized animal agriculture? Um,
Yeah, yeah,
yeah.
yeah.
There's a great question here from Sean that thanks you for a really phenomenal presentation, and asks whether your lab has investigated the strength of attachment between the bovine cells and the decellularized spinach. So have you assessed what % of cells attach versus how many cells just kind of remain in suspension without actually adhering?
Yeah, we haven't done that yet, but we are literally in the process of that. And so, yeah, I greatly appreciate that question. So, what we purchased one of the BIDI systems that allows you to, it's kind of a microfluidics device, but you can control the shear stress over the cells. And so you can determine, how much shear stress for how long is applied so that the cells, will essentially come off. And so we're currently doing those experiments, and hope to have that wrapped up in a few months. But yeah, we're
Awesome.
excited about those experiments. I am, at least. I don't know if my students are, because it's technically challenging work, but.
Yeah, and there was a question here, I think that's related, since you mentioned your students. Is the center of gravity of this research still at WPI? Oh, or has it moved with you to Boston College?
It's both. I mean, it's I've moved to Boston College, but a lot of my collaborators are still back there, and so I think it's really at both. And um, I made so many wonderful connections in my time at WPI that as long as my colleagues are there, part of the work is going to remain there.
Yeah.
So yeah, I think it's really between both places.
Awesome. Yeah, it's so cool to see these multi-institutional collaborations form, especially as they relate to alternative proteins.
Yeah, and of course we're right down now. I'm right down the road from Dave Kaplan, so he's a good friend and colleague. So hopefully that Boston Worcester corridor is going to be strengthened.
Absolutely. Too cool. Here's a question from Forrest. Do plant scaffolds allow fluid flow both into and out of the structure like veins and arteries do? Or like veins versus arteries?
Right. Yeah. No. So it turns out that unlike our veins and arteries, they're basically one way. And so what we're looking at to create the return flow is taking one leaf, putting another leaf on top of it, and having one inlet flow and the other one outlet flow. Some of the other things that we've done in the past, and again, this is more because it adds cost, and so this is probably more useful in human tissue regeneration, is as the plant leaves are growing, you can actually stack two of them on together. You can kind of fix them together so that they then grow together as one. And my colleague Tanya D'Amico and Pam Weathers back at WPI, they showed that after allowing them to grow together like that, fuse together, the arteries or the veins essentially from one leaf will connect to the veins to the other, and so then you can create that flow pattern that Forrest was I think asking about. But it is more user intensive.
Yeah, very cool. That's a clever workaround. There's a question here from Yao. You saw that the cells from different cows might result in slightly different results. So did you check to see if the different varieties of plant sources might also impact the final product? And I assume they're talking about, where you source your spinach from and not spinach to broccoli variation.
Yeah, no, and that's something that we were surprised to see quite the difference between the cow one versus cow three essentially. We were really surprised with that, and so it wasn't until we saw the data then we started questioning, okay, what are the differences? Could we get more information? Unfortunately, we didn't have any more information on the cows than where we received the cells from, and what muscle. So we don't know why. It could be the spinach leaf too. There could be differences. We buy the leaves all together in one batch, but that doesn't mean that they come from the same plant and same field even. So there are a lot more questions that arise than we've answered. Yeah.
Yeah, sounds like, as we look to scale up this process, we'll need to think about controlling for batch to batch variation. In much the same way that, the plant-based meat field is kind of struggling with today, right? Like where they source their plant materials have a really big bearing on the end product and functionality. So very cool. Are there physical and or chemical characteristics that can be used to determine if a new plant material is a good potential candidate for seeding cells? So beyond the ones you've tried, what are the criteria that make a plant material a successful cell seeder carrier?
I think probably the most important thing is the topography of the plant. And so once it's decellularized, because I think that's really going to help the alignment of those cells. And again, that'll probably be really important for taste, that texture. In regards to, we don't know if there are other, physical and chemical characteristics. One of the things that actually a high school student started doing for their high school science fair project about a year ago, and unfortunately COVID hit, so he wasn't able to do it to the same degree he wanted to, but basically decellularize meat. And let's look at the extracellular matrix that remains after it's decellularized in meat. What are its mechanical properties? And then can we match that with a plant? And so he was able to do a little bit of this at his house, really, which was pretty impressive. But can we actually take a good scientific look at decellularized meat and compare it to decellularized plants, mechanical properties, the topography, all that there. A lot of opportunities there.
Yeah, it's very cool that was a high school research project, because it does seem like this line of research inquiry, at least the first phase of it, in which you're decellularizing the plant material, seems to have really pretty low barriers of access. Like you had your recipe right there on a single slide, and it seems like it's an opportunity to crowdsource, like decellularizing all of the materials in the plant kingdom. Like all of us, the 50 of us left in the seminar right now could just pick one off your short list and see.
A few years ago, a master's art student over in England did her project looking at like 100 different plants and flowers that she decellularized and turned into this amazing work of art. So yeah, I think there's a lot of different opportunities, but again, she did this herself and she wasn't a scientist, and so I do think there's a low barrier to entry.
Do you have like a short list of plants, fruits, vegetables that you want to do this research with and haven't yet had the time? Like what's on the wish list?
Well, I don't know if it's a list as much as the very first plant that we tried was a tomato, and
Hmm.
we failed miserably. I'd love to be able to get back to the tomato and do it again on a tomato and make it work. But yeah, that's um, I guess one of the other plants I'd like to try is going back to our cardiac research. Heart valves are made of leaflets that are very thin, and so when I look at basically a blooming flower like a rose petal, it's very similar to what I think of as a heart valve leaflet. And so I think it'd be really interesting to look at that and see how it compares and will it function as a leaflet, a heart valve leaflet after it's been decellularized.
Ooh, that's really cool. Or like an artichoke, I think follows a similar type of structure with those, but slightly thicker. Um,
Yep.
yeah,
Yeah.
that's so interesting to think about. Um, so there is a question here about whether or not you've looked into existing plant waste products or agricultural waste streams and utilizing those for scaffolds.
Yeah, that's one of the things we're starting to look at right now. And again, with our colleagues at Wisconsin, and looking at corn husks and things like that there, which already have an aligned structure to them. And so, yeah, I think that's a definite possibility and something that we really want to explore further.
Awesome. Um, a question here from Jonathan Avasar. How mechanically robust are the constructs? So can they actually be manipulated by hand?
Most of them, yes. They, especially once you get the cells on there, they start off kind of like a wet tissue paper. And so, if you know,
Um,
yeah, you want to be very careful with it. But once you start putting the cells on there and you get the cells laying down a little bit of extracellular matrix, it stiffens it up a little bit. And so it becomes a much more, pliable and workable.
mm,
Yeah. So I would say it's on the border. It, they literally, once you hydrate them when they're lyophilized, they're freeze dried, they're nice and stiff. But as soon as you hydrate them, it's like a wet tissue paper. And if you fold it over on itself, you've got to be careful peeling it back off.
mhm.
So
Very cool. Um, I like this next question from Isaac as well. Does the seeded spinach still have the kind of like crunch from the cellulose in the plant? And if it does, how do you think you would get around the texture issue if what you want to do is stack these three-dimensional structured whole meats? Like, will it end up just being a really crunchy steak?
I think you don't need to add the spinach to your steak burger anymore. It's already going to be in there.
Mm,
So I don't know. I don't know if it'll still have the crunch or not. I don't think so, based on the mechanics that I've felt. But we haven't actually done a shear experiment to determine how much force is required to shear through that and compare it to actual steak.
mm,
Um, but some of the things that we had thought about in the past are, should we add cellulase, which would break down the cellulose more? And so there could be some opportunities there to, use some enzymes to help digest the cellulose to give you that, you know, more compliant structure if that's what you're looking for.
mhm. Very cool. There's a question here from Paul about whether you can tell us a little bit more about your cell sourcing. From which muscle groups did you collect your cells and animal age or type?
Yeah, not off the top of my head, but that's all. We just published a paper a couple months ago in Food Bioscience, which is open access. And so, if you want to email me, I can. My email is just Glenn GLENN dot Gaudette GAUDETTE at bc.edu, and I can forward it to you. I can attach it. But it's in food bioscience, and all that the information should be in that paper there.
Awesome. This is from March 31st, right?
Yes. Yep.
Okay,
Yep.
I just dropped a link to that paper there in the chat, so you all can read it. Thanks
Thanks.
for making that open access. Yeah. Awesome. There's a question here from Nicholas Guys. Thank you for a very beautiful, very elegant talk. This might be a bit off topic, but what are your thoughts about optimizing the cell lines you use with genetic engineering? Do you think that there are any advantages would, you know, advantages from a technological perspective of using this approach would be offset by the limited product acceptance by the public?
Yeah, that's my biggest concern is, how will the public feel about it? And so I worry that there will be a group of people who are going to be completely against cultured meat, and they are going to use anything they can to limit the field and limit the growth and try to really, discourage people from eating cultured meat. And so that's my biggest concern. However, I do think that there are going to be a lot of opportunities. And one of the things I'd love to see us being able to do is produce more food for the world, right? And I think the genetically manipulated cells are probably going to be better. They're going to proliferate faster. They're going to differentiate better. They're going to be a better cell source. And so I do think there's going to be an opportunity for that. And I personally am not that concerned about, the genetic manipulation in terms of eating it. So I think there's a potential for it. I just worry that when it's first introduced, I hope, I just worry that if there are too many flags, that the public will not accept it, and it'll take longer for the public to accept it. I think the public's going to accept it, one day. I mean, look at, the plant-based burgers, and veggie burgers, and they, came out a long time ago, and, it took a long time before people would, accepted those. And now, I mean, the Impossible Burger, is everywhere. That's fantastic, right? So, yeah.
Yeah, it's really hard to predict the kind of long arc that consumer acceptance sometimes takes. I think
Yeah.
especially with food, because evolutionarily we're kind of hardwired to be skeptical of new foods.
Right.
Yeah,
Yeah. I made a meatloaf recently out of the Beyond Meat that I thought was, really, really good. I don't
yeah,
know. I don't know what my family thought about it, but I thought it was
I did just, as you were responding, it it occurred to me that a really good set of explorations for a social scientist to take on in parallel with technological advancements in cultivated meat, plant-based meat, and then agnostic of whether or not there's a genetic engineering component, is like, how do we get the general public to be less apprehensive about these tools, that the cut and paste tools that we have as kind of a privilege of modern day science? Like, how do we chip away at the consumer part of that and make it a less scary prospect, not just as it pertains to food, but kind of at large? This embracing the best available tools that society has available to us to solve the big problems we're faced against.
Yeah, and that's why I think it's so important that we work with, others from outside of our field, right? Outside of the conventional space that we talk about.
Mhm. Yeah, I love the like true multidisciplinary web that you painted for us in some of your last slides.
That's what I believe in. So I've learned so much from my colleagues that are outside of the technical fields. It's amazing, and it's made me a better engineer, and it's made me a better person. So, um, if you'd asked me 10, 15, 20 years ago, I probably wouldn't have thought the same, but I've experienced it. So.
Yeah, super cool. I do think it's just like infinitely valuable to continue to learn new frameworks for thinking about the same kinds of questions.
Yep.
Yeah. Well, there's one last question here. in the chat, and that's kind of revisiting your slide about the case for cultivated meat. Are there any like technological factors, to your knowledge, that make it impossible to create, meat that fully replicates the sensory and functional experience of conventional animal meat using plants as opposed to animal cells?
Um, I think it's going to be getting the actual protein content without using the animal cells could be tricky. And I think getting the exact same kind of texture of the striated meat, you know, is it going to be close? Can we make it close? I think we can. Can we make it, can we make it acceptable? Yes. Will it be the exact same? I don't think so. I just, so the food connoisseur maybe could tell the difference, and so then becomes a question of really, at what point do you tell the difference? So, for
Mhm.
example, wines, right? I can't tell the difference between, a very, very expensive wine and, a table wine, right? Other
Mhm.
people can really, they really, really can. I think it's going to be similar. Um, and so, part of it is, what are we aiming for? And if we can, the bottom line is we've got to replace as much of the conventional animal agriculture grown meat as we can. And so I'm 100% in support of
Mhm.
plant-based meat. I think that's fantastic, 100% supportive of it. But I'm also a realist, and I know that there are people that are never going to, try it and never accept it.
Mhm.
Um, will cultured meat be acceptable to them? I don't know. I hope so. It's the same thing as real meat, you know.
Yeah.
So, I hope that we can convince them.
Yeah, absolutely. When you take into the like very complex social and cultural attachments people have to meat, the way we associate it with wealth and power and status, I think there is a real case to be made that plant-based meat, cultivated meat, the products and fermentation all have a role to play in this future protein supply. Um, yeah, well, Glenn, that was the end of the audience questions for you today. This was a remarkable seminar that I'm excited to watch back again and again because some of those graphics were incredible. And it's so cool to see how your work has evolved over time. Um, I found so much value from this. I know our community is really grateful for you taking the time to share your knowledge with us. Before we wrap up today, are there any final thoughts you would like to leave our community of scientists with?
Well, I just want to say thank you to you and to Good Food Institute, and all you're doing for the field, and to all the scientists and non-scientists out there. This work is so important. This field has been so collaborative. I hope it continues, to work together, and to embrace all the diverse thinking, the diverse people that are involved in the field, and continue to grow. Um, you know, Everybody is making a contribution to this field, and we should be grateful for everybody's contribution. I certainly am, and so I'm appreciative of everything that everybody's doing, and just want to ask them to continue the great work that they're doing.
Thank you so much. Yeah, I think you did a great job of ending off your presentation also with basically a million white spaces that still exist, a bunch of unexplored questions that we have yet to tackle. So, like you were saying time and time again, there's ample room for everyone to get involved. For those of you who are interested in learning about specific white spaces or unexplored questions in our field, you can check out one of our open access resources at gfi.org/solutions. And maybe reach out to Glenn if you have further questions about his research or his work growing the ecosystem at Boston College. Glenn, it was so fantastic to have you here. Everyone, always really fantastic to have you join from every corner of the world. I'm really excited to invite you all back again next month for a wonderful seminar from our our friends at Penn State University. So Glenn, it was really fantastic to have you here again, and I hope you have a fantastic rest of your week.
Thank you. Take care.
Thank you. Bye.